The second type of acetylcholine receptor are the nicotinic receptor. As you remember, the nicotinic receptors are also receptors having affinity to the acetylcholine. They also have affinity to the muscarine, but their affinity is high to the nicotine and low for the muscarine. And taking all together, the muscarinic receptors as the nicotinic receptors have approximately the same affinity to the acetylcholine. There are two types of nicotinic receptors. The first one is called N1 or NM because it is present at the level of neuromuscular junctions, and the second one it is called N2 or NN, and it is found in the autonomic ganglia, in the central nervous system, as in the adrenal medulla. The nicotinic receptors are receptors that act by activating an ion channel, and therefore they belong to a ligand-gated ion channel. And I do mean by this, when the nicotine is present, it binds to the nicotinic receptor, and it causes a conformational change in this receptor, leading to an opening to the sodium channel, allowing the entry of the sodium ions inside the cell, resulting in the depolarization of the effector cell. When it does this action, it does it when it is provided at low concentration. But when the nicotine is provided at high concentration, then the nicotinic receptor will be blocked. Which one specifically? The blockade of the nicotinic receptor is specific to the drug and to the location of the nicotinic receptor. For example, the mecamylamine is a drug that blocks the nicotinic receptor at the level of ganglia, while the atracurium blocked the nicotinic receptor at the level of the neuromuscular junction. So now, after explaining the cholinergic neuron and the receptors of the acetylcholine, now we're gonna mention what are the drugs that can replace or act similarly as the acetylcholine. Those drugs are known to be cholinergic drugs, and usually these drugs affect the autonomic nervous system, and they act on the same receptors activated already by the acetylcholine. They are classified into two types, direct acting as an indirect acting. And we will be seeing in the indirect acting, some of them act in a reversible manner, while others act in an irreversible manner. To start, by the direct acting cholinergic drug or cholinergic agonists, why they are called so? They are called direct acting because they mimic the action of the acetylcholine itself in a way that when the acetylcholine is found normally in the synaptic cleft, itself it binds its receptor on the postsynaptic membrane. When we talk about a cholinergic agonist, if it is capable to replace the acetylcholine and sitting on the same place, then it will be a direct acting. And this place can be muscarinic or nicotinic receptor. Usually the direct acting cholinergic agonists have a longer duration of action than that of the acetylcholine. We can classify them into two groups depending on their source. Some of them are endogenous and others are alkaloids naturally occurring. The endogenous choline ester are the acetylcholine and others that are synthetic ester of choline, such as beta-nicole and carbachol. The naturally occurring alkaloids are the nicotine and the pilocarpine. I'm gonna start by the endogenous neurotransmitter made by the cholinergic neuron, that is the acetylcholine. We know that the acetylcholine binds to a receptor present on the pre or post-synaptic membrane. This is because the acetylcholine itself does not have the capacity to penetrate a membrane. It is made by the cholinergic neuron present in the parasympathetic division, in the somatic nerves, as in the autonomic ganglia. The acetylcholine usually is a neurotransmitter that lacks therapeutic importance. And this therapeutic importance is let's say, is lost or is reduced because the acetylcholine is made by many types of neurons in many locations. So we can use the term multiplicity of action. And this multiplicity of action makes its effect or the effect of the acetylcholine very diffuse. This is one. The second reason why the acetylcholine lacks its therapeutic importance is that its high ability to be degraded by the acetylcholinesterase. The acetylcholine usually is not too much stable, so it is rapidly inactivated by the cholinesterase. And as I mentioned before, the acetylcholine has both activities, muscarinic and nicotinic. Now, regarding what does the acetylcholine exert in our body, I'm going to cite some functions of the acetylcholine. First, the acetylcholine is involved in decreasing heart rate and cardiac output. How it does this? Usually, its action on the heart mimic the effect of a vagal stimulation. Remember that the vagal neuron is the longest nerve of the autonomic nervous system, and it is a neuron that helps to regulate many critical aspects of human physiology, including heart rate, cardiac output, blood pressure, and digestions, for example. So when the acetylcholine is injected intravenously, it causes a decrease in the cardiac rate. This is what we call negative chronotropy. And it causes also a stroke volume as a result in a reduction in rate of firing at the SA node. And we know that the activity of the normal vagal neuron is by releasing an extra acetylcholine at the SA node, causing the reduction of the heart rate. The acetylcholine is involved also in decreasing blood pressure. How it does this? The injection of acetylcholine into the vessel causes a vasodilation and lowers the blood pressure. How it does this? The acetylcholine, once it is injected, it binds an M3 receptors that it is found on the endothelial cells lining smooth muscle of the blood vessels. This binding activates a G protein that activates by itself a phospholipase, inducing the activation of many endogenous enzymes, leading finally to the production of nitric oxide from L-arginine. This nitric oxide diffuses into the vascular smooth muscle cell and stimulate a protein kinase G production. This kinase G production inhibits a phosphodiesterase that cause the conversion of the GTP into cyclic GMP, that it is involved in the stimulation and hyperpolarization of the smooth muscle cell, leading to a relaxation of it. In the absence of administered cholinergic agent, so suppose that we do not administer a cholinergic agonist, what does it happen really? So usually it is not mentioned yet that the vascular cholinergic receptor may exert certain function, and their function is not yet known because the acetylcholine is never released into the blood in significant quantities. to block these muscarinic receptors and to prevent the acetylcholine from producing vasodilation in certain patients, we administer an antidote of it, which is known to be the atropine. The acetylcholine exerts also many other functions, such as it increases the secretion of the salivary gland by the production of saliva. It stimulates also the peristaltic movement of the intestine, stimulating its motility and secretions. It enhances the bronchiolar secretions. It increases the tone or the contraction of the detrusor muscle surrounding the bladder, causing them a stimulation of the urination. It stimulates also the ciliary muscle contraction for near vision, and it is also involved in causing miosis, so by the contraction of the pupillar sphincter muscle.